Starting? Okay. Do we have
Yeah.
good enough audience? Yeah, I don't see.
Now that almost 20, let's see, 21 have joined, I think
Okay.
we'll be joining. We'll start. Yeah.
They will be
Okay.
joining. So
Yeah.
Okay. All right.
Good morning,
So
all. Let me give you give an introduction and then we'll start.
Sure.
Yeah. So good morning, all. Good morning, ma'am, and good morning, Robin sir, Arunan sir, and all the participants who have joined. So this is second of the series of webinar that we are organizing under Kerala Biotechnology Commission of Kerala State Council for Science, Technology and Environment. For your understanding, ma'am, Kerala Biotechnology Commission is the agency under the government of Kerala, which is mandated to strengthen the biotechnology in the state of Kerala. So as part of its awareness programs and all, we frequently conduct some webinars, seminars, etc. So today we are delighted to have with us for this insightful session on pharmaceutical development, end-to-end and patient care QTPP towards safer, faster and smarter therapeutics. We have with us today Dr. Anuji Abraham, an accomplished pharmaceutical executive with over 14 years of leadership experience, spanning end-to-end drug development, product strategy, and innovation-driven therapeutic design. Dr. Anuji Abraham currently serves as the Senior Director and Strategic Leader at Alta Sciences USA, where she spearheads product development initiatives that bring together scientific depth, operational agility, and patient-centric design principles. And her work focuses on translating patient needs into smarter, faster, and most impactful development pathways, an approach that is at the heart of today's webinar theme. So before joining Alta Sciences, Dr. Anuji held key roles at Bristol Myers Squibb, contributing across material science, engineering, and drug product development leadership. Her career spans a rare blend of technical expertise, cross-functional collaboration, and strategic oversight across global teams in pharmaceutical ecosystem. And she earned a PhD in physical chemistry from ETH Zurich, Switzerland, one of the world's most prestigious scientific institutions. Over the years, she has authored more than 40 peer-reviewed publications, reflecting the depth and impact of her scientific contributions. And beyond these technical strength, she's admired for her passion for mentorship and scientific leadership. She's deeply committed to nurturing young professionals, strengthening research culture, and fostering collaborations that accelerate the journey from lab to patient. So today, she'll be sharing her expertise on pharmaceutical development end-to-end and patient-centric QTPP towards safer, faster, and smarter therapeutics, highlighting how quality target product profiles anchored in patient needs can reshape the development strategies and streamline regulatory alignment. So all participants, please join me in warmly welcoming Dr. Anuji Abraham to lead today's session. Over to you, Dr. Anuji, ma'am.
Thank you very much, Sarika, for the introduction, a kind introduction. And, time here, I'm dialing in from the US, time here is 10:30 p.m. I was telling Sarika that I'll do, for Kerala, for a talk in Kerala, I will take 10:30 p.m. Yeah, so thanks to Sarika and Arunan for the...
So much, yeah.
So first of all, thank you, Sarika and Arunan, for a kind invitation. I've known Arunan for, I don't know, the past about 15 years, I would say, Arunan, right? We had an overlap at EPFL while I was doing my first postdoctoral research at EPFL after my PhD at ETH. I'll walk through the career journey, and thank you. Nice to see you, Arunan, again. It is exciting to connect back. So my talk, as Sarika was telling, it's going to cover the pharmaceutical development end-to-end and just giving some elements into patient-centric product development, or QTPP, the quality target product profile. So which is very important, in terms, it's not just developing the product, but you're developing the product for the patient, and patient has to be in the middle of, in the center of this development. So I'm Anuji Abraham, again, Senior Director and heading pharmaceutical development at Alta Sciences in the US. So just to give an overview of the talk, so the outline, and I'll like to talk, I believe, most of you are students who I am talking to. So I would like to give a little bit about me and my career path. It is always good to see, where people come from and where people are, where is your journey taking you, right? So for instance, I was thinking, Arunan had his, done his PhD in India, I guess, then, postdoctoral research abroad. He decided to move back to India, and Sarika has taken a career in India. But for me, I moved out and decided to stay outside, moving from Switzerland to UK to US. So everyone has a path, and this is a science path, right? It's not just, in India, we have the notion that we have to be doctors and engineers, but I'm not a doctor or an engineer, doctor as a medical doctor. But you have a path. You can, that is something, we should be seeing, like people like Sarika and Arunan and Robin here and myself, who has taken this journey through science, not being an engineer or a doctor, but pursued science, pure science, and taken a career. Yeah. So I'll talk about my path, then a little bit on the pharmaceutical development phases. So when you're talking about the development, discovery, all the way to the launch of a product, discovering a molecule to the launch of a drug product, it is, going through different phases, right? It is the phases are characterized by the clinical stages and taking it to the launch. So we'll talk a little bit on that. And I want to introduce, this presentation is going to be the basic fundamentals, right, of development. So what is drug substance? What is a drug product? And what are the different molecular modalities which is involved in the drug substance and product? We'll talk a little bit about that. And then we can talk on the patient centricity and how quality target product profile is centered around patient's need. And at the end, I would want to touch a little bit on the artificial intelligence, or AI, and automation in drug development. So how we can leverage it, not to be scared of it, but really embrace it and leverage and to accelerate drug development. So this is me. So my expertise, I have done a PhD in NMR spectroscopy, actually, in material science. Did a lot of application there. Then, so I would say, starting from NMR spectroscopy to material science to drug delivery, my expertise span across NMR, materials to product development. I've done 40 publications over my career. Not, usually that's again one of the things I want to emphasize, is that if you want to publish, you need not have to be in academia, right? You can be in industry and continue publishing. That's something else you can think if you want to go into the scientific path, the career, even in an industry. Some of the interest, as Sarika was telling, I'm passionate about mentoring. I continue to connect with university students here in the US. I am an outdoor person, so I like running and biking and anything like that, and doing things. I have 2 kids, so I do a lot of outdoor stuff with my kids. Yeah. My mantra is actually learn the industry you are in, no matter what your role is, right? You can be a scientist, you can be a data analyst, you can be an IT person. If you are in the pharmaceutical industry, learn the industry, right? So that helps you to give the context of why you are there, how you can impact, right? So that is the fundamental to wherever you are. You can be in pharmaceutical industry or engineering, civil engineering industry, I don't know, wherever you are, right? Whatever your role is, learn the industry you are in. And some fun facts, I've lived in 3 different continents. I was born in India. I had the opportunity to live in Europe for several years doing my PhD and postdoc. And then I moved to the US, and I'm living currently in the US. I also authored a kids book, which also talks about career in science as a woman. That's an illustrated kids book, which is published on Amazon. Yeah. So as for the career path, did my PhD in ETH Zurich, which was in NMR spectroscopy and applications in materials. Then I did 3 postdocs before spend a little longer time in academia before I joined Bristol Myers Squibb, which was my first role in industry. So I did a postdoc in EPFL, then I went to Durham University in the UK, and then to WVU in the USA, joined Bristol Myers Squibb, and I was there for 14 years before I joined Alta Sciences US. In 2025, September of 2025, I made a move to join Alta Sciences as a head of pharmaceutical product development. During my time at BMS, I also went and did a mini MBA. I wanted to get a little bit of understanding of the business side of things, of the industry, and from London School of Business in the UK. So when you talk about pharmaceutical development phases, it starts from discovering a molecule, right? This is on the very left-hand side. If you see my mouse moving, you can say you start with finding a molecule, right? That finding one molecule start with several thousands of molecules. So you are screening one molecule to get to this preclinical stage. So this stage from exploratory discovery to getting to full discovery and preclinical, you are narrowing down the number of molecules which you think was a hit to get to that one molecule, or perhaps two as a backup to get to the preclinical stage. So once you have a molecule identified which is potential for a certain target, you have to, before you get into humans to do anything, right, you need to prove that this medication is safe, that it's not toxic, right? So you do what we call preclinical animal studies.
Excuse me, Dr. Aniji, I think the presentation shows two slides together. I mean, not picture-in-picture style. I don't know how it happened.
Can you see now?
Yeah, it's okay now. It's perfect now. Yeah, it's okay now.
Okay.
Yeah, very good. Okay, thank you.
Okay. No problem. Tell me.
Yeah, okay, it's okay now. Oh, it's
okay now?
again. No, again the picture-in-picture. Okay, fine. Yeah.
Now it's okay? Okay.
It's okay now. Yeah.
Okay. So so where were we? So we have, from discovery to preclinical, you're getting to one molecule, and that has to be tested in different species of animals. So you go up in species, right? You start with a rat or mouse and going up to dog, which you think it's closer to, depending on, not every model will work for everything, right? So you go through the models to get some understanding into the toxicity and some of the PK, the efficacy aspect of the drug. Then after that, you go to what we call the phase 1, which is first in human. That is the very first time the drug is introduced to a human being, right? And it takes months to years when it goes from phase 1, you call phase 1, 2, 3, and 4, then you have the launch, and still you continue doing some of the trials which we call as life cycle management. So it takes, from this identification of a molecule all the way to the launch, it is typically 9 to 10 years time, right? That is the pharmaceutical, the standard in pharmaceutical industry. But that's shrinking. And I can tell you some of the reasons why it's shrinking. And it shrunk very much for COVID drug, which was an accelerated case where things get, approved super fast, right? Because it was a need, it's an urgent need for the whole world. And so that is an exceptional case. But typically, it is 9 to 10 years, but it's reducing to about 6.5 to 7 years using some of the things which I will touch upon later on. It's mostly like accelerating through AI, the clinical trials, how AI and automation can be used in several stages of development. Yeah. Okay. So the next So let's start with the fundamentals, right? There is drug substance, which is which is the molecule. The active pharmaceutical ingredient is the drug substance, which is responsible for intended therapeutic effect. That is the molecule which is giving some sort of effect in your body, for the indication you are working towards. Then there's the formulation. It is the process of combining the drug substance with ingredients, which we call excipients, to create a stable, effective, and safe medicine. And finally, you have the drug product, which is what you see when you buy a medicine, right? And which is the final formulated medicine, which combines your active pharmaceutical ingredient, which is an API, with excipients at specific dosage form. So that is your drug product. So these are the fundamental things you have to remember. There's the drug substance, the formulation, and getting to the drug product. I am So emphasizing again, what is a drug substance? It's an active pharmaceutical ingredient. It is the active molecule. It can come in any molecular modality. It can be a small molecule. It can be a complex molecule. It can be a large molecule. We'll get into that. What does it mean, right? And as per FDA's definition, a drug substance is intended for use in diagnosis, cure, mitigation, and treatment or prevention of disease, for use as a component in the medicine, but not in a device, So it is a component in a medicine, but not a device. That is the definition of a drug substance as for FDA. So as I mentioned, drug substance can be defined in different ways. So the way I'm going to define it is through molecular modalities. So there is small molecule, right, which is molecules which are less than 1,000 Dalton. Then you have millimolecules, which is something in between. You have peptides and siRNAs and mRNAs. All of that comes into this category. And then you have the biologics. They are very large molecules with molecular weight in about 100 kilodalton, which is the proteins and monoclonal antibodies and antibody drug conjugates. All of that will come into that category. So those are the three main buckets of drug molecular modality for a drug substance. But what is a drug product? Product is the final formulated medicine combining API with the excipient. So it can come in various forms, like tablets, capsules, suspensions, injections, nasal, rinse, name it. There's a lot more. So these are a few examples. So the key aspects, the criteria for a drug product is it has to be stable, it has to be safe, it has to be manufacturable. Manufacturability is a key aspect. I'm in the product development. So the formulation and the manufacturability for designing the drug while designing the drug product is important. You can have like a novel drug product. If you cannot scale and manufacture it, it can't be a product. And the efficacy of the product. So these are the key criteria to make a drug substance a drug product. So there are different types of drug products. So this is not, any modality can be anywhere in terms of molecules, right? You have the oral products, the injectables. The orals are the tablets, the capsules, solutions, and suspensions, which you give to infants. Injectables can be intravenous, subcutaneous, intratumoral, IVT. There's so many different kinds of injectables. Then there are the others I call, which are the topicals, the transdermals, inhalables, all of that. So each of the molecular modality can come into any of this delivery, right? A small molecule can be oral, Milla can be oral, injectable can be small, oral, and biologics. So this, anything can happen, but this is the definition of product by the delivery. And back to the formulation. So getting from substance to product, you need the formulation. Formulation is the process of combining the drug substance with ingredients like excipients, we call, to create a stable, effective, and safe medicine. But it defines the composition of the active ingredient and excipient. So if you are looking if you're making a cake, you have the recipe, right? So this is the recipe for your getting to your drug product. But it's not only that, the compositional aspect, but it also captures the process and the process of it, right? Sometimes some things need blending and roller compaction, and there is a huge process to mix these API with excipients, and each of the excipient has a function to it. And so, and there is a process, and the process has parameters involved, and that brings the drug product process. The formulation captures both the chemical composition, the process, and the parameters of the process to make it to a drug product. So why does formulation matter? Again, emphasizing on the formulation development. It's not mixing things, right? It is important. Formulation is key. In thinking about the formulation is key to develop the right drug product. Getting from a drug substance to product. So for instance, taking, you ensure precise dosing, enhancing the solubility, permeability. There has to be need. Sometimes you have to enhance your solubility or permeability through formulation process. Enabling targeted delivery. Sometimes you can have excipients which targets to, you make certain, for instance, taking a liposome, right? You have your drug substance and encapsulated in lipids, which could have targets to it, which helps you to target to specific, cells, right? So formulation helps in targeting as well. So for instance, giving an example, a poorly permeable drug, it needs a different kind of formulation. It's not, again, mixing things. It needs a specific formulation. For instance, another example is extended release. You can have something which releases in your stomach fast or releases in your stomach with time, right? Meaning that the absorption in your stomach is with time. So at the end of the day, the drug has to get into your systemic, circulation, which is your blood, right? From thinking about orally available drug products or drug substance. So you can, your formulation for an extended release versus an immediate release will be very different. The excipients you would use for these two would be very different in order to get to that immediate to that sustained release or extended release. So it is a very key element for the formulation development is a key part for drug product development. And that brings us to a high-level definition of TPP, which is the target product profile, which it helps to develop the formulation that is safe, efficacious, and easy for patients to use. So what are we working towards? You are working to make a drug product, and what should your drug product look like? What is that goal, right? The TPP is going to tell you that, right? How is your delivery? How often should I have to take this medicine, right? Is it once a day or once a week? All of that is defined in your TPP, quality target product profile, which will help you to design that drug product, right? So it's helping you to design, to make ultimately a stable, safe, efficacious drug product, which is manufacturable, as I said. But another element which comes into is the patient centricity, which is not captured here, which I'll come to it later, but just want to throw it out there again. The patient centricity aspect is a key, central of all of this has to be the patient needs, right? And there are regulatory guidances in terms of, even in terms of quantifying, patient centricity, it's difficult to quantify, right? But FDA has guidances in terms of how you can semi-empirically quantify how much of patient centricity is involved in your design, right? Or the final product, yeah. So a little bit more into the drug molecular modalities and the products. So let's dive a little deeper. So for small molecules, starting with initially I showed a slide with 3 different molecular modalities: the small molecules, the intermediate molecules, and the large molecules. So let's start with small molecules, which are very small. The molecular weight is less than 1,000 Dalton. It is structurally, they're very simple. They can be chemically synthesized, that we can make it. You have a, reaction to make the, a pathway to make to that product, the drug molecule. It commonly, these, small molecules it's orally taken, right? It's tablets or capsules, you see, but it can be injectables, inhalants, or topicals. But these are all the advantages of having a small molecule, right? It is easy. It's easy to handle. It's stable. It's usually stable at room temperature, making it easy to store and distribute, right? For instance, when we buy capsules and tablets, it's easy for us to store. You do not have to put in the refrigerator. Everything is easy. And it typically metabolizes in the liver and excreted in kidney. But one of the biggest cons for small molecule is that it's not specific. Even though at the early stages of development, you are looking at, these molecules to fit into certain targets, but there are similar multiple targets in your body. If you are aiming to get a specific target, that specificity is lacking for small molecules, meaning that it will target multiple places that will lead to more side effects, right? So that is one of the things with small molecules, which we can mitigate through more targeting using molecules. And there are ways you can conjugate a small molecule to biologics and target it. There are ways we can make it more specific, but small molecule as such is less specific. So a little bit more into the small molecules. If you think about a small molecule, you are thinking about molecules which you are taking orally, most of it. That there is a classification system called BCS classification. It's called Biopharmaceutics Classification System. It's a scientific framework that categorizes drug substance based on their solubility and permeability. It's not only that your drug substance has to be soluble in your body or in your stomach and intestine, but also it has to absorb. The permeability is also a key piece. So the larger the small molecule is, the larger it's fun, historically, if you think the small molecules were 250 Dalton, right? Now the small molecules are becoming bigger, up to 700 Dalton. So it's becoming big, and that makes it difficult for these molecules to permeate through membranes to get into your systemic circulation. So it's classified. There is class 1, which is the best case scenario, right? When drugs started, most of the molecules were in this category, class 1, where it was highly soluble and highly permeable. Then now we are in a scenario where most of our molecules are class 2 and class 5. Class 2 is low, which has low solubility with high permeability, which is still okay. It is low soluble. We can use some enabling to get it more soluble. And there is class 5 molecules, which are both not soluble and low permeable. So they are like rocks, right? It's hard to deal with such molecules. So if you look at the approved drugs, 60% of the approved drugs come in this category of 2 and 4. So these drugs would need some sort of enabling, either for solubility or for both, for solubility and permeability. So that's something you have to keep in mind. So molecule is not a molecule. There are the physiochemical properties of the molecule is very key to get the right formulation, to get the right drug product, right? So yeah, that's what I was highlighting. So just a little bit more into that material selection, right? So you have a small molecule. So you're thinking of a molecule in solution, you crystallize it out into some form, which we call as the free form. The free form can exist as different polymorphs, right? Different crystal structures. They are different polymorphs of that molecule in solid state, and it can be milled. So something when you grind, you are enhancing the dissolution, right? You are increasing the surface area of these little particles to get it dissolved faster, right? So you're increasing the dissolution, right? And sometimes that's not sufficient, right? That is not helping. So you need to look into other ways how you can increase your solubility. The other approaches are you can make co-crystals. You can make your API crystallize with a co-former and make co-crystals, or you can make a salt of your API. Sometimes some of the drug products, when you look, you can see that it's not, it's the HCl salt of something, right? Salt of something. So that is how you enhance the solubility. So inherently, using co-crystals or salt, you are inherently increasing the solubility of the molecule. The other way is you can go into amorphous dispersion. Anything you make amorphous system we call as solid solutions. So you're making kind of a solution in solid state, which makes it easy to disperse and solubilize. So that is one other approach. One of the biggest problems of amorphous drug is that it is metastable. It can easily recrystallize. So you don't want things to change when the drug is sitting in a, when your drug is in a tablet, right? So there are ways you can circumvent that by making this amorphous dispersion in a polymer matrix, which, stabilizes this amorphous system in a polymer matrix, and you can, enhance the solubility, but also keep the stability of the molecule. And then there are ways you can, you need formulation to enable, right? So in that case, think about a BCS class 4 where solubility and permeability is inhibited. In that case, we might need some sort of components you add in your formulation to enhance this permeation of the drug. So there are excipients we call permeation enhancers, which are components in your formulation or the drug product, which helps to bring along your drug molecule into the systemic, cross the membrane, right? So there are ways you can circumvent that. Sometimes you make some sort of micelles with your drug molecule and this permeation enhancers, which helps you to move the drug into the systemic circulation. So these are the technicality, the nuances, the fun part of drug development. So, just in terms of the drug product, which comes from the small molecule, it can be, as I mentioned, it can be tablets, capsules. Once you figured out, okay, you try to enable the maximum as possible as a drug substance, then you formulate and make the drug product. Mostly in this case, in small molecule case, it's tablets, capsules, suspensions, and injectables. Mostly it's tablets and capsules. So now the next category is the millimolecule, which has molecular weight about 1,000 Dalton or greater than 1,000 Dalton. So the biggest advantage for those systems are those molecules, the high specificity and selectivity for the target, which means low toxicity, and they are biodegradable. And the main cons of this is that the poor oral bioavailability. If you try to take a peptide or semi-glutide, you have heard of Ozempic and Wegovy, right? So the insulin, all of those, if you take orally, it is easy to digest it, right? So it won't keep its form. So its bioavailability is very poor. And another thing for it is it's very difficult to cross the membrane. They are very large molecules, and it becomes very difficult for the molecule to cross the membrane. When larger the molecule, it's difficult to cross the membrane to get to the systemic circulation. So low permeability is one of the things, shorter half-life, and it is often has low stability. It is prone to hydrolysis, oxidation, etc. And typically we need a complex formulation for this, more complex than a small molecule formulation. So some of the drug products which comes out of millimolecule molecules are mostly parenterals, meaning injectables, subcutaneous, intravenous, all of that comes from millimolecule. And sometimes it can be tablets and capsules as well. A lot of companies are trying to develop peptides as tablets or capsules as well. You have to circumvent some of the degradation aspects, and you have to overcome the barrier of the bioavailability as well, right? By enhancing the permeation. So using some permeation enhancers in the formulation. So the last category is the the large molecules, which has molecular weight about 100 kilodalton. They are very targeted, right? And so because of that, they are very targeted. They have reduced systemic side effects. That is the biggest advantage of going to biologics. They have longer duration of action, right? Sometimes you inject and you need the injection every week or every month, sort of. The cons is that it's not orally, you cannot make an oral product out of biologics. And typically infusion or injection is required. That is, not as convenient as taking a tablet or a capsule. High cost involved with this. The development of biologics is extremely expensive. And typically, even to store these products, it's very expensive because it needs refrigerated conditions. So some of the examples are monoclonal antibodies, bispecifics, proteins, and antibody drug conjugate. So this is a scenario I was telling you can conjugate a small molecule drug to a monoclonal antibody and make it targeted. So for instance, you can have a targeted delivery of your small molecules by going through this conjugation step. So again, emphasizing on some of the products which are coming out of biologics. Most of them are in injectables, can be intravenous, subcutaneous, intratumoral. Sometimes it could be patch pumps in the auto-injectors or patch pumps. You can have pumps which continuously, pumps the drug into your body. So that there are more innovative ways of making that again comes to that patient centricity aspect, like making the life of people, if you have to go through injections every 2 days, maybe using a patch pump might be the better way, from a patient point of view, right? So companies try to develop these kind of devices, product in device, in order to enhance that experience for the patients. So now let's dive a little bit into the patient centricity of the QTPP, quality target product profile, for the drug product development and design. So this is where, I want to emphasize again, quality target product profile plays a central role in regulatory submission, right? If you have a story to say that, these are the things we have taken care to make sure it is patient compatible, there is, your regulatory pathway is easier. The QTPP is foundational for the quality by design, for the design of the product. And it also guides the identification of the critical quality attributes. These are measurable properties that can be controlled to ensure the product meets the QTPP, so the CQAs. And it's a living document. It evolves as data and knowledge, as we gain data and knowledge, right? Again, QTPP, the big umbrella is that, when you design a product, it has to be efficacious, safe, stable, and manufacturable. And all in center will be the patient. We are always working, making a product for the patient, and the patient centricity should be center for the design of the product. So a couple of examples, right? Very simple examples, just so that it can stay in your mind, is the development of an extended release tablet. Let's say a scenario, you have to take a patient has to take 2 tablets, 3 times a day, right? So that is something you have to remember, and it's too much of inconvenience if you think, right? You have to carry around. If you have your little vial with these tablets or capsules which you carry around to take the medication. So instead, you can think of having sustained release, meaning that you do not have to do every 2 tablets 3 times a day. Instead, you can have tablets once a day or once every 2 days. So in order to get that, you need to, taggle with, kind of figure out your formulation in a way that the formulation will provide a sustained release. So, if you look at your plasma concentration over time, if you have immediate release, you have these spikes. Whenever you take a drug, you spike, right? The plasma concentration, the drug concentration in your plasma goes up, and it goes up and up and up, right? So you can think of a formulation where instead of going up and up, you are gradually releasing it, right? You're gradually, so the drug can have some sort of, can be in some sort of matrix, some sort of excipient where it releases some amount in the stomach. Then it has a certain amount in the intestine. So it prolongs over time, which helps you to maintain a certain level of drug in your plasma concentration, which we call as extended release. This is again thinking around the patient centricity, right? So you are making the life of patient easy by doing, reducing the number of doses the patient has to take. That's one small example. And this is an example which which is easy to remember. This is the development of the insulin pen. If you think the starting in, from 1920s to 1980s, how insulin was injected into your body. So initially it was all the glass syringe. We use glass syringe to inject. It was painful and it was difficult, and the needle size was big, and, and then when it, with time, companies heard what the pain the patient has to go through this while going through this process, right? Then it became plastic syringe. Then it became an insulin pump, which you carry around. Now you have the insulin pen. It's like, sleek and nice, and, it's easy, and you don't even feel the pain, right? So how the product or the device evolved for the product over time is a very good example. This is a very good example of how the device, the product, including the device, has evolved around the patient, right? So this is Novo's, the display is actually Novo's easy to use insulin syringe here. So before I finish my talk, I don't know, how is my time? Yeah, we have time for Q&A, right? So just wanted to give a little bit of flavor in terms of AI and automation in drug development. So let's go back to our whole, the phases in drug development, starting from the discovery all the way to the launch, right? So just giving you some snippets of where, where it's heavily used. So one of the main places machine learning models are used is at the very early stages of discovery, right? There was many companies, small companies, developing this platform where you can, instead of making thousands and thousands of molecules, you are, designing your molecule through this machine learning models to get that hit. When I say small molecules, when you are going and targeting certain proteins in your cells, you have a certain construct for it, right? And the models, like the chemists are thinking, right? The models can make it faster and avoids making less number of molecules. So you are narrowing down the synthesis of fewer number of molecules, likewise accelerating this process of identifying that first hit, right? So that is one place which is heavily used. Larger pharmaceutical companies are leveraging, either building internal capabilities or partnering with external, companies where they have these platforms to accelerate drug development. That is one place. The second place is, really machine learning models are used to forecast pharmacokinetics, toxicity, and efficacy, helping to select molecules with optimal profiles before laboratory testing. So when it comes around preclinical studies, right? So you can use models to accelerate things. Then there are, clinical trials. Robin is on the call, so you are more on the clinical side of things, right? So how, some of these AI tools can help identifying suitable participants, monitoring the outcome in real time. All of that can be done. Automated data analysis, right? That is automation, not AI. Automation existed even for a long time, but now we are emphasizing all of this after the birth of AI, right? So that is another place we can heavily use. And robotics and automated systems are used for quality control, scalability, formulation development. It's so much used in formulation development and manufacturing, continuous manufacturing, the automation aspects around that. Then also for the regulatory data, right? Facilitating the compliance and accelerating the process of approval. So I've given like snippets of things, right, across this different phases, starting from discovery, where ML tools are heavily used, all the way to getting to the filing, right? But how do you prepare, right? You are graduates. I guess those who are listening this call are mostly science graduates. So how do you prepare yourself? You are thinking that, oh my, AI and automation is going to take over my job, right? No, it's not. So it's building the literacy, right? Gaining experience with digital tools, embracing that interdisciplinary collaboration. So you are subject matter expert with deep technical and scientific depth of expertise in a certain field, right? Can it be formulation, making a drug, whatever that is, right? You have that. The critical thinking and adaptability are the key things, right? Don't be cocooned in what you're doing. Connect with, understand where efficiency can be brought in, right? Through AI tools and automation. So which means that collaboration, right? You might not be a data scientist, but a pharmaceutical instance, you're joining a pharmaceutical company as a scientist with science degree, you have deep technical expertise, some sort of literacy around AI tools and how this is used will help you to communicate clearly with the data scientist to provide that value proposition in order what to be achieved, where can things be efficient, right? Those sort of conversations can happen only if you have certain level of literacy in it, right? So AI is not going to replace you. It's only augment. It will not replace. So the scientific expertise, keep it, and the critical thinking are key things for the next stage, right? I didn't have to think when I came out of my PhD, I didn't think of any of those, right? When you come out as a master's degree or PhD or whatever you're getting, these are the things you can keep in mind with your science degree. With that, I think that's my last slide. So I'll let people to ask questions. So this is where I'm concluding. This is a very high level overview of things. If you want to dig deeper into anything, feel free to reach out to me. I'm happy to talk, on specific topics, especially for students, right? And thank you for attending, and listening in.
Thank you so much, Dr. Anuji ma'am, for that informative and inspiring session. It was indeed, you have made everything so simple. I thought.
No. It is, we should think, that it is simple, right? It's not complicated. When you think from a, like.
It was indeed a great session. Great to listen to you. And now I invite all the participants. You can ask questions. You may either post your questions in the chat box or use the raise hand option if you wish to ask any questions, please.
I don't see any questions in the chat.
Yeah.
No questions, but I really enjoyed. It gave me some ideas. Maybe when I talk, I can tell the part 2 of the story.
That'll be fantastic. I am going to listen into your talk, by the way.
No, I was just wondering, like, what should I talk about? And this gave me some idea. So maybe part 2 of the story is a good topic. Taking from evidence generation in humans to more about data augmentation and health economics and outcomes research, real world evidence, all of those things. Maybe
that'll be fantastic. Yeah. It will be definitely a continuation, right?
Yeah.
The
students.
students.
Can I ask one question, Dr. Beniji? That was a wonderful talk.
Of course, yeah, please.
I thought it was mere chemistry, but you actually skipped chemistry, and it was a wonderful talk. And I heard this kind of personalized medicine. What exactly? Can you talk something around personalized medicine?
Yeah, so personalized medicine is more, cell therapy is one example, right? Where you're harvesting cells from your body, right? Depending on what you, and you modify, right, the cells and put it back into your body, right? So that is a simple way to describe, right? It's either a gene therapy or cell therapy, but you are making the drug. For instance, we have, BMS used to have cell therapy pipeline, right? Where you harvest, from X person and you give back to that person, right? And that is personalized because the whole manufacturing you are doing for one patient, right? So that makes it extremely expensive, right? So by one dose or one time introduction, you are basically curing the disease, right? So that is the benefit of it. But one medication becomes, personalized if you think it's easy for certain people, the affordability of that, right? It becomes like, it's a philosophical thing as well, right? Yeah, you can make personalized medicine by harvesting cells through cell therapy or gene therapy, but it is very expensive. Yeah.
Yes, thank you so much. And I have another question. Usually it takes a lot of time to market a drug, really. So usually, how much time usually a drug need for getting to market? 10 years or more than that?
Yeah, so it's about 9-10 years, right? But
Okay.
now pharmaceutical companies are thinking of shrinking that, right? Through this, implementation of ML models and automation, making things efficient, faster to get it to 7 years, right? That is like the blue sky scenario. Every company is working towards is shrinking the timeline from 9 to 10 years to 6.5 to 7 years, right?
Okay, good. Okay.
Yeah, that is the accelerated path.
And another question is, we heard that some drugs are withdrawn from market after a certain time. So why it's so? And why this problem? There will be some problem associated with that for withdrawing the drug. So why it's noted before it come into market?
So that is a very good example of life cycle management, right? You are always, so some things, as you said, I don't know which drug you're specifically mentioning about, right? But you could have everything going fine, but suddenly you see some sort of reaction in patients sometimes, unfortunately, it could be after the launch, right? In such scenarios, you might have to pull the drug back, right? So that is why, the life cycle management of the continuous monitoring and continuous evaluation of the data is important even after the launch of the product, right? So yeah, that can happen, yeah, because it's like, when you are going through clinical trials, you are looking at some effects on patients, right? And instance, during clinical trial, if something adverse event happens, even if sometimes it might not be drug related, but you might have to stop because of the adverse event, right? You cannot pursue with that trial because an adverse event happened. So the safety aspect is key, right? So at whatever stage that is, yeah.
Got it. Thank you so much.
Yeah. Further along you go, you're thinking things are getting safer, right? Yeah.
Ma'am, I have a question. Ma'am, Kerala gives more push on towards nutraceuticals these days, and many products market in the name of nutraceuticals. So, what's your take on that? Does that require a clinical trial, or in the name of food? I mean, food that doesn't require a clinical trial. No?
Yeah.
So, would that be?
I mean, that's a completely different industry, to be honest with you. The regulations are different, right? So it's an easier path, right? It's if you want to get something out there, but a lot of times, the other way can also happen. The nutraceutical route can go into pharmaceutical route, right? So I don't have a specific, it's a completely different industry, right? It's a completely different market, right? For nutraceuticals. It's easier to get things out there, right? As a nutraceutical than a pharmaceutical.
So a few questions are there in the chat box.
Okay.
Okay, ma'am. Mary, if she wants to, you talk about a little bit about a life after PhD and your journey towards to a senior director.
Oh, so life after PhD. Okay. So, as I was telling early, right? I mean, we have multiple scientists here sitting, right? Sarika, Arun, and Robin, myself. We all took different paths, right? So either life after PhD can be, you can be inspired to go back to your country and have positions there, right? Or you decide to stay here and pursue a career within the country where you are, right? So when I did my PhD in Switzerland, right? And I knew, it's a good place as a university. It was great. I interacted with brilliant minds. It was wonderful time I've had there. But I always wanted to move out and live in an English-speaking country, to be honest. So that was my motivation to move out and go to the UK first, and stayed there for about a year. And then ultimately in my mind was getting to the US, because research is more more in the US. It happens, I feel, right? Compared to European, you have more fundings. Now things are changing, but when I moved, it was different. Okay. Now today is a different scenario. When I moved, it was more money was there in the US for research, right? So it was easier as a scientist to make a path here, right? And once you get all honesty, I can tell you, once you get your PhD, things are easier in the US. All your process of, this is not a time for me to talk about immigration, but, all your green card and citizen, all of that is so simple and straightforward. You should have the label of PhD. That's it, right? Everything becomes expedited for you. So I think Robin will agree with that, right? So it's not difficult, right? And that's how I felt. And when I moved here, first thing I can tell you was I wanted to be in academics, right? The reason I did 3 postdoc was to be in academics. But academics in the US is extremely competitive, in the sense you have to apply for all over the US, right? But my personal life also came into play, right? At the end of the day, I wanted a family at some point, and I wanted to settle down, and I restricted my area to northeast, right? Which is New York, New Jersey, all of this. So you are kind of confining, and where I ended up has the most number of pharmaceutical industry, right? So that motivated me to apply to a pharmaceutical company, and it just worked out, yeah. And I think, getting that first step, having a PhD at that time helped, right?
Thank you. And another question: how efficient are computer biology, bioinformatics, and math to model to predict how drugs interact with biological systems? I think you have probably answered.
That is, yeah. I mean, I don't want, sorry, let me see the question again. Computer biology. Yeah, I mean, the computational science is used, right? When I said machine learning modeling, right, which involves also a lot of molecular dynamics, when you say computer biology, is that what you're mentioning? All modeling tools are used along with, right? So it's not just the machine learning, but we also do computational modeling along with machine learning to come up with these hits. And sitting with the chemist as well, right? Chemist with tremendous amount of experience in terms of they can look at a molecule and say that, oh, this should fit here, right? Sort of. So it's like brains plus the machine learning plus the computational model. Yeah. So yeah, yes, we can. The short answer is yes.
So yes, that's done with the questions. No more questions. Shall we wind up?
Okay, yeah,
So,
for sure. Yeah. It was such a pleasure,
yeah.
I don't know how many students, I couldn't see the number here, but I hope you
Yeah.
benefited some from this talk, right? And
Joy.
feel free. Yeah. And if you have specific questions, feel free to reach out. Sarika has my email, and so is Arunan. So I'm happy to answer anything further, even in terms of your career or anything you want, right? I'm happy to chat. Yeah. Thank you again, Arunan and Sarika. It was such a pleasure seeing you again, Arunan.
So,
Yeah, that's true.
And-
Thank you so much. Thank you so much, Halini.
Yeah. So as we come to the end of today's session, I would like to extend my heartfelt gratitude to Dr. Anuji Abraham for being with us in this late night there, for sharing your expertise and valuable insights. Thank you so much, ma'am. It was indeed an engaging discussion. So my sincere thanks to Arunan sir for connecting with Anuji ma'am. Thank you, Robin sir, for joining us. A special thanks to all the participants. I hope you all enjoyed and had a great takeaway session from today's session. And we hope today's webinar provided meaningful insights into the patient-centric pharmaceutical development. Thank you all once again, and we look forward to your participation in the future programs of Kerala Biotechnology Commission. Have a wonderful day ahead. Thank you so much.
Thanks again. Bye now.
Bye.
Yeah, great.
Bye.